RNA polymerase II core promoters in eukaryotes. We all know that RNA polymerase, in order to initiate transcription, has to bind to a region that is called as a promoter region. Now, the eukaryotes having three major RNA polymerases, polymerase 1, 2, and 3. Every polymerase seems to have its own promoter sequences or promoter region. And so in this session, the focus is going to be on the core promoter region that helps the RNA polymerase to bind to the gene uh, that it has to transcribe. So let us look at the learning outcomes of this session. The RNA polymerase II machinery needs a core promoter. That is something that is very clear. And this core promoter is generally a contiguous stretch of DNA sequence. And within this contiguous stretch of DNA sequence is present several specific cis elements. And these cis elements are the regions where you can have the transcription machinery assembling. So that is another important thing that one comes to know. Then, unlike in prokaryotes, where the promoter is always upstream of the gene to be transcribed, in case of eukaryotes, the core promoter sequence not only encompasses the transcription initiation site, but it has the specific cis elements being present upstream of the start point of the transcription and also downstream of the start point of the transcription. So that is why it is over an area around the transcription start site, unlike that in the prokaryotes where the promoter is only upstream of the start point of the transcription. There are observed to be two types of core promoters for RNA polymerase II, and that is focused core promoters and the dispersed core promoters. Focused core promoters are considered to be ancient. When you say ancient, that means that may have come first, and from these focused core promoters may have evolved the dispersed core promoters. But the understanding between the two is that this is considering only a very short stretch of DNA sequence within which you have the start point or the start points present. That means you can also have multiple start points over here. But in case of dispersed, it's definitely multiple start points, but not a very short sequence of DNA. It is across a broader sequence of DNA. The various cis elements that have been found associated with several RNA polymerase II core promoters are the TATA box, the BRE, the INR, MTE, DPE, DCE, XCPE, etc. Important to note is that many of these have also been associated with control of regulation of transcription. So many of them would help in deciding whether transcription has to begin or doesn't have to begin. Now let us look at or compare the focused core promoters with the dispersed core promoters and thereby understanding the structural implications. So if you have a gene of interest that is expanding, so if this is the start point of the transcription of the gene, okay, then one would find that it is within this particular region where you have all the required cis elements present for transcription to get initiated. So you can see that the transcription is initiating at a very focused region. So you may either have a single start site or you may have distinct clusters of start sites, but it is always present within a very short stretch of nucleotides. When you compare that with the dispersed core promoters, again you have the gene that has to be expressed and you will find that it is not as focused. So you will have a long stretch of DNA containing multiple start sites from which you can have the transcription initiation beginning. And this can range anywhere from 50 to 100 nucleotides. And also interesting to observe is that these have been found to have what is called as a CPG islands. And the dispersed core promoters have been maximally present in the vertebrates. In fact, in vertebrates, you will find that there are much more number or a larger proportion of promoters being dispersed core promoters rather than focused core promoters. In humans, you have both focused as well as dispersed core promoters. So, the focus core promoter, in fact, has been very extensively studied, and a lot of several key specific elements, that is, the cis elements, have been found to be associated with the focused core promoters, and they have been studied extensively with also understanding what is the sequence, what is whether it has consensus, whether it is conserved, so on and so forth. In case of the dispersed promoters, what has been noticed through various studies is that many of the dispersed promoters are Tata box less promoters. So, in fact, one of the motifs that is present in the core promoters called the Tata box is not found in the dispersed promoters at all. This is something that is has been importantly noted, because when you compare it with prokaryotic promoters, in the prokaryotic promoters you will always have a Tata box present. Okay, and so therefore that you know a concept that you can have a promoter without a Tata box is there in the eukaryotic system. So let us look at how these core promoter elements are positioned within the promoter region. So we are now going to consider mainly the promoter region, and within the promoter region you will find a motif that is called as the initiator. And interesting to note is that this initiator is anywhere from -2 to +4 relative to the start point, which means the start point is present within the initiator region itself, and noticeably the start point is always an A. Upstream of the start point, which is within or encompassed by the INR, is present another motif, which is called the Tata box, and it is present within -31 to -26 region upstream, and that is why it is -31 to -26. And this Tata box is known to have a consensus sequence of TATAAA, which is the reason why it is called as the Tata box motif. Again, upstream of the Tata box is found another cis element, and this is between -37 to -32, and this is known as the BRE. Now, before we go into what is BRE, please understand that in many core promoters, one also has a BRE element just downstream of the Tata box. So you can have a BRE element upstream of the Tata box, and you can have one downstream of the Tata box. So these BRE elements are able to bind to the transcription factor 2B. And interesting also to note is that these, unlike the Tata box, which is a AT-rich region, the BRE is a GC-rich sequence. Now we come to a motif that is present downstream of the start point. So here it is very clear that the core promoter elements in eukaryotes can be present both downstream as well as upstream. And so the promoter elements is actually around the start point of the transcription. So here this motif that is present between +28 to +32 is called as the downstream processing element, and this downstream processing element may be preceded in many promoters by another cis element which is called as MTE, or if there is no MTE there may be present what is called as DCE, and DCE is present even upstream of the MTE per se, but of course it is downstream of the initiator region. Now, both the DPE and the MTE or the DCE are able to bind to transcription factor 2D complex or related proteins. So this is how you can see that all the elements of the core promoter, of the focused core promoter, is present within -40 and +40 of the start point. So that is the region where you have all the specific sequences present, and so you can say that it is a highly focused region, and that is why it is called as the focused core promoter. Now one goes to the Tata box, looking at it in a little more depth than we have already looked at. So Tata box was the first core element to be discovered in eukaryotes, and its consensus sequences has been found to be TATAAA. But there are a lot of variants of this Tata box motif. So you may have a lot of different sequences of the TATA box present in different species, or even within a cell in different cell within a species in different cells. Different promoters can have different sequence of the TATA box. It is present upstream of the gene at around -30, as we just observed, -31 to +26. And the position can vary in different species. So it's not, we do say that it is around -30, therefore, because the position in different species can vary. It has been correlated to the Pribnow box, which is a TATA box that is present in the prokaryotic system. You can take note of the fact that the Pribnow box has a consensus sequence of TATAAT, and this consensus sequence in the eukaryotes is TATAAA. So therefore, there is a lot of resemblance between the two. But then eventually, over a period of time, there have been many differences that have been found with respect to the Pribnow box. So the Pribnow box in the prokaryotes is positioned at -10, whereas you will never find a Tata box motif in the eukaryotes positioned at -10. It is generally around -30. And over and above that, again, you can have even from the -30, the position being variable. So that is something that is important to note. Now, the TATA box is bound to by a protein that is called as TATA box binding protein. And interestingly, this TATA box binding protein is part of a complex that is TF2D. So you can see that TF2D could bind to DPE-MT, but it can also bind to the TATA box, which is present the upstream of the initiator region. Other than TBP, what is also able to bind to the TATA box is what is called as TBP related factors. There are several TBP related factors like TRF1, TRF2, etc. So the humans also have been found to have TRF2 and them being able to bind to the TATA box per se. Now, Tata box is not really found in vertebrates, and most of the vertebrates have been found to have dispersed promoters. So, therefore, the idea or the conclusion that the vertebrates which have dispersed promoters are Tata box less promoters. That is, they don't have the Tata box. Interesting again to note is that even within the focused core promoters, it is not necessary that you have to have a Tata box present in the promoter region. So you cannot say that the Tata box is universally present in all focused core promoters. In fact, what has been observed is that the percentage core promoters that have Tata box vary from species to species. So say for example, you have the humans who have a certain number of focused core promoters, and even within the focused core promoters that it has, only a certain percentage of those core promoters actually have a Tata box motif. So therefore, it being present or not being present depends on the promoter itself and the gene that it is associated with. But what has definitely been observed is that Tata box as a motif is associated with the focused core promoters. The next in line is the initiator. This is a this is a region, or this is a motif that is encompassing the transcription start site. This is present whether it is in the dispersed core promoter or whether it is in the focus core promoter. It always encompasses the transcription start site. It has been present in promoters that have Tata box and also present in promoters that don't have Tata box. So there is no relation with whether the promoter has a Tata box or does not have a Tata box. So independent of the Tata box, the INR can be present. And most studies have shown that it is actually the INR which is the most common motif that is present in most focused core promoters. This initiator region is bound to by the TAFs that in fact are associated with the TF2D complex, and the TAF binding to the initiator has found to be sequence specific again, thereby keeping this fact that the initiator would have a consensus sequence. So in humans, the consensus sequence is pyrimidine, pyrimidine. Here mostly it is the cytosine. A plus 1, it is written as A plus 1 because this also is the start point of transcription. Any nucleotide over here followed by an AT or another pyrimidine and another pyrimidine. So you can see that this portion is conserved, but then you can have these pyrimidines keeping changing. So there can be a lot of variation from the consensus sequence, and yet there is a consensus sequence. In the Drosophila, the sequence has been observed to be TCA plus 1, which is again the start point of transcription, GT or TCT. So you have to keep this in mind that within the INR is always present the start point of the transcription, and that is generally the A. It is always an A. So you have this A actually conserved in the INR sequence. Now this that is being represented is basically a single start transcription point, but you can have multiple transcription start points as well, and then that means that you would have several INRs present. RNA Pol 2 can also directly bind with the INR, or it can bind through the transcription factors. Now TF2D complex is definitely able to bind to all the different variants of the INR that are present. And the INR binding by the TF2D is also observed in the dispersed promoters. So therefore, this is in a way suggesting that INR can be present in the dispersed promoters. In fact, since they are TATA-less, the INR presence is considered to be important in the dispersed promoters. Then we go to the next core promoter element, which is the BRE. So the BRE basically is transcription factor 2 binding response element, and the TF2B effectively binds to the BRE consensus sequence, and this binding is considered to be sequence specific. So this interaction of the TF2B with the BRE is very important for assembly of the pre-initiation complex. So the consensus sequence, as you can observe over here, is a GC-rich region with the 5' end of, the 3' end of the BRE immediately positioned before the 5' T of the TATA box if the promoter has the TATA box. This is something that has been observed in many core promoters. And as noted earlier and mentioned earlier, you can have the BRE present upstream of the TATA box and also downstream of the TATA box. Now, looking at the consensus sequence and its interaction with TF2B, what has been found is that this sequence, that is the BRE, may be responsible for either positive control or negative control on transcription initiation. And a part of this has been attributed to, if you have promoters having BREU and BRED, then the consensus sequence of these two are different. So therefore, the binding with which the transcription machinery binds may differ based on what is the consensus sequence, and thereby you may have a positive or a negative control of the transcription initiation. But point to be noted over here is that BRE has a role to play in control of transcription regulation. Then we come to DPE and MTE. The downstream processing element binds to the TF2D, and it has been important for basal transcription. That means binding of the DPE to TF2D has been observed to initiate transcription. MTE stands for motif 10 element, and it is present downstream at around plus 18 to plus 27 relative to the start point of the gene, which is again encompassed by the initiator region. The consensus sequence at about plus 30 can bind specifically to two proteins, which is called as TAF6 and the TF9, that are associated with the TF2D. There are studies that suggest that TF6 and TF9 have what is called as histone folds present. So they have specific motifs present within their structure, and this histone fold protein enables it to bind to the downstream processing element. So this has been correlated to the histone binding to the DNA. And uh, 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 several studies have also importantly shown that the DPE actually works in tandem with the initiator region to initiate transcription, and the distance between the INR and the DPE has been found to be a critical component of actually deciding whether transcription is going to be weak or it is going to be strong. The MTE is functioning also cooperatively with the INR, and in this case also there is a strict INR MTE spacing requirement. MTE, however, can compensate for the loss of the TATA box or the DPE. So, if you have a promoter in which the TATA box or the DPE is removed, the MTE can hold the responsibility of enabling transcription to begin. Then we come to DCE and XCPE1. DCE is downstream core element, and it is divided into three sub elements. So you have S1 with a consensus sequence of CTTC from +6 to +11. You can see it is downstream of the start point of transcription. S2, which is CTGT from +16 to +21, and the S3 AGC from +32 +34. This is closest to the downstream processing element, that is the DPE. Now all these three have been able to or have been shown to bind to TAF1, which is again associated with the TF2D complex. XCPE1 stands for X core promoter element 1, and this is present from -8 to +2. Again, relative to the start point +1, which is being encompassed by the XCPE1, just like the INR did. And so XCPE1 also has a consensus sequence, and it has been found to be working with activators in CPG islands. So XCPE1 is definitely associated more with the dispersive promoter than with the focused core promoters. So let us make the concluding remarks. RNA polymerase II core promoters are varied in different species, with some being focused core promoters and the others being dispersed core promoters. The focused core promoters have very specific motifs, which are cis elements. So they are basically sequences, nucleotide sequences, which in turn are going to help in transcription initiation. The motifs have been associated with core promoters such as TATA box, the BRE, the INR, the DPE, and the MT. These are motifs that are associated with the core promoters. And most of these motifs are binding sites for various transcription factors that help in marking the position where the pre-initiation complex is to assemble. Therefore, the core promoters of RNA polymerase II contain specific cis motifs that are present both upstream and downstream of the transcription start site, and that enables transcription initiation. So they are helping in beginning transcription by the RNA pol 2. And with these points or motifs also being points of regulation. Thank you.